All right, hello everyone. My name is Erik Lamoureux, and I'm a PhD student in the MAL lab. Today I'll be discussing how to assess red blood cell deformability using deep learning. So red blood cell deformability is an important biophysical property that determines the cell's ability to squeeze through and navigate the body's microvasculature. Transfusion red blood cell deformability varies based on both the donor and the blood storage time. The sorting of blood based on deformability would allow for patient-specific allocation of transfusion blood, where for chronic transfusion patients, they should receive long-lasting or more deformable blood to increase the time between needed transfusions. And for acute transfusion patients, they can receive short-lasting or more rigid blood, as they just need a blood top-up and can normally produce an adequate blood supply. Here, I present a deep learning approach to determine blood deformability that is accessible to anyone with an adequate optical microscope. So here I'll describe our general approach. A red blood cell sample from a donor is processed and infused into the microfluidic device at 1% hematocrit. The cells are sorted by their deformability levels and donor rigidity score is determined. After sorting, the cells are removed from the device and placed in imaging well plates. Using a Nikon microscope at 40x magnification, full well image scans are obtained. These full well image scans are passed through a custom computer vision segmentation algorithm and are augmented by rotating the images to produce balanced classes of different deformability levels. The training dataset is used to train a convolutional neural network which learns relevant image features to detect the deformability of red blood cells using new and previously unseen images. Finally, the deep learning derived deformability score can be obtained and is compared to the microfluidic method. So a previous PhD student in our lab developed this microfluidic ratchet sorting device. Red blood cells are passed through the device to assess their deformability. The cells are squeezed through progressively narrower tapered constrictions, so more deformable cells pass further through the device to lower numbered outlets, while more rigid cells are filtered more quickly to higher numbered outlets. Here shows the microfluidic deformability-based sorting results. This plot displays the cumulative distribution of the sorted cells. We observe donor-specific deformability patterns, and these are quantified at the 50% crossover frequency, termed the donor's rigidity score, or RS. So here on the left, we see example images from outlets 2 to 5 for our third donor. These images are input to the deep learning network to learn the relationship between cell morphological features and deformability. The middle shows the training, validation, and testing accuracies of our deep learning model for differentiating between soft and rigid red blood cells for 10 donors. And finally, on the right, we see a visual representation of the morphological features that the model identifies to be related to deformability. So overall, the deep learning method was applied to 10 donors with an even sex split and for a variety of blood types. The training data for all 10 donors converged, and testing accuracies ranged from 68% to 98%, with an aggregate accuracy of 84% plus or minus 11%. The deformability curves and rigidity scores varied between the donors, and as seen from these plots, the deep learning model successfully predicts the donor's rigidity score using data separate from its training. The aggregate average and rigidity score difference between the microfluidic and deep learning methods was 0.13 plus or minus 0.08. Finally, the deep learning rigidity score is plotted versus the microfluidic rigidity score. We find high convergence, R equals 0.93, between the rigidity score evaluations using the two methods. So where do we go from here? We have a mouse model study in the works where we look to track transfused cells using fluorescent labels. Blood draws are taken from the mouse at regular intervals post-transfusion, and the samples are put through the microfluidic device. We hypothesize that more rigid transfused cells are preferentially cleared, so over time more fluorescently labeled deformable cells will remain relative to the more rigid labeled cells. Results from the mouse study will inform us on the feasibility of using deformability as a biomarker for transfusion lifetime. This will be further investigated in a clinical study with myelodysplastic syndrome chronic transfusion patients. Samples of transfusion blood will be taken and microfluidically sorted, and the rigidity score of the bags will be related to the time between subsequent transfusions for the recipients. This will inform us about the relationship between red blood cell deformability and transfusion lifetime for MDS patients. Finally, the deep learning deformability prediction method will be applied to donors while their blood is stored. We aim to evaluate how well the computer model can detect storage-related morphological changes. If the deep learning model can be made more robust, it'll greatly expand the access of red blood cell deformability evaluation to any lab or clinic with an adequate microscope. Thank you for listening. I would like to thank all the people, research groups, and funding that made this work possible.
No, it's okay. They're about to bring you a microphone.
Nice presentation. Which mouse strain are you going to be using for the study, since not all mouse strains store and circulate the same?
Yeah, that's a good question. I think they're like, I don't know exactly the strain of mouse. I haven't worked on that project in a little bit now, but Dr. Ma would know. Sorry about that. Black 6? Is that okay? Black 6 strain.
Thank you. That was really interesting. I'm Tina Jacobucci. I'm the team lead of transfusion services at St. Paul's. And I'm actually pretty blown away with all of the information that's coming out about. And so coming from a clinical perspective, we are all about inventory management. And what I'm hearing is this just adds an extra layer of finding compatible units for recipients. So for the sort of microscopic testing that you've been doing, how long does it take? And how complex would it be to train a basic lab technologist to be able to do that sort of thing?
Yeah, so that's a great question. The pipeline for doing all this isn't at a stage where it would be really accessible for introduction into clinical groups just yet. One major limitation of our work is that we only find that we can get a model to predict the rigid or soft cells based on donor-specific training. So you can't train the model yet on a bunch of donors and then get it to spit out a accurate deformability prediction on a new donor. It has to be trained on the specific donor that is being tested on. So that's one major limitation. And right now, the main bottleneck in applying this is in the data processing side. It takes a lot of time to take the data and segment the images into single cell images for use in the model, and it takes hours or sometimes days to manually audit that process to make sure we have quality data. So with improved methods for image segmentation, this could be done a lot faster, and with a larger data set of a lot of donors, we expect that it should be able to generalize to new donors. And the implementation of that, when it's at a stage that can be demonstrated, should be relatively straightforward. And yeah, could train a lab technician on it with ease.
Just further to that, couldn't this technology be applied earlier? Wouldn't Canadian Blood Services just love to do the testing as these donor units came to them, and they do the provide that information to the user in the hospital, et cetera, and somehow rate it for whatever use, just like they do for other bloods. Just throwing that out.
Yeah, certainly. One thing that we want to do that'll help the accessibility of this approach is to see if we can get the model to learn deformability based on blood smears. So it's just a standard, laboratory practice. And if we can do that and integrate the use of this model into existing procedures, that would also help its implementation across the board.
That's great. Thank you very much, Erik.